I'm recording. I'm struggling to remember everything. Sorry, people. Right, from this slide, you can see that we need light microscopes and electron microscopes. And as I said, today we need electron microscopes to see viruses, DNA, molecules, proteins, and even down to atoms. But because of the limitations of an electron microscope, which can't see living things, we can't follow live events, we can't always determine what protein we are looking at. We can look at the virus, or we can look at the protein, and we can look at the organelle, but it's not always easy to see what it really is because it's full of information. And with a light microscope, especially fluorescence microscopes, you can identify specific proteins based on the light that you've, or the dye that you've added to that protein. So light microscopes have their space, but the problem is they don't have the resolution of an electron microscope. So people, as you know, scientists are never ever really happy. So they would always try to break the limits, and that is where super resolution microscopy comes in. So if I put the next slide in, if you look at a piece of, I think this is tubulin, this comes from a paper in Journal of Cell Science. If you look at, I think, tubulin, and you look under a confocal microscope, you can't see the thickness that we can see here. You would basically see this tubulin as thick as this blob here on the left-hand side. Just imagine this is the thickness of the tubulin. Obviously, we are not very happy with that. We would like to see tubulin much finer, especially because it comes in networks. We don't see it only in their single form. So when you use the next super resolution, the SIM type of super resolution can bring down your resolution to about 100 nanometers. And then this tubulin would look more or less like this thickness. If you go to another type of super resolution called STED, you can bring down the resolution to about 30 to 70 nanometers. But then still not really what we would like to see. The third type of super resolution on the market, PALM or STORM, or the new name they call it is SIMLIM. That brings down this resolution down to even 10 nanometers. If your system and your sample prep and everything works perfectly, theoretically you can get down to 10 nanometers. This is just an overview type of slide. I can give you the reference to the paper. It's a very, very nice summary of the current super resolution systems on the market. So what is super resolution? Why is it important? Why is it there? The first person, or the person whose name is coming up every time we talk about resolution, is Ernst Abbe. Now, Ernst Abbe actually has a very he was involved in the foundation of ZOYC. He and Carl ZOYC worked together, and with the ZOYC people, Anya and Sune might even give you some nice background on him and history. But basically, he was the person who first described resolution, not only in a nice definition, but also with a mathematical equation. There's this monument for him in Jena, which is the headquarters, I think, of ZOYC. And I'm not promoting ZOYC, I promise. This is the person who everyone else after Ernst Abbe is still using his theories and his explanation of resolution. So what is resolution? Resolution is the resolution of an optical microscope is defined as the shortest distance between 2 points on a specimen that you can still distinguish as two separate entities. If we look at the diagram here on the right hand side, d-min means the limit of the shortest distance. So if the distance between 2 points is further than the minimum, then you can easily see them as 2 points. However, at some point, they reach that point where you can just about see, okay, there are still 2 points. We call this the Rayleigh limit. But then if those 2 points move closer together, you can see these yellow bumps. If they move closer together, you will not be able to resolve them anymore. They will just look like one blob of fluorescence or light there. In a confocal microscope, what this means is that you need the smallest distance that you can see two molecules apart in the X and Y axis is about 200 nanometers. In Z, unfortunately, that's a bit less easy to resolve. That would be about 500 nanometers. So what we are looking at is the glow. I'll talk about this point spread function soon, but basically each fluorochrome that you have in your sample has a glow around that fluorochrome. And that glow we call the point spread function. If you would put the point spread function on a little graph of intensity, this would be the intensity plot around that glow. And in X and Y, 200 nanometers. In Z, this would be 500 nanometers. Just get next. Right. So this point spread function, what it is, etc. It's basically, you would have a fluorophore right in the middle of the circle, but you won't ever be able to see that with normal light. Maybe with an electron microscope if you know what you're looking for. But in terms of the fluorescence that it emits, it will always be a larger visualization of light. So this will be a circle in X and Y, and in Z it will be this elongated shape. A different way to show it is on this way. It's very similar to this previous image I showed. If you present that this point spread function as a function of its intensity, it's most intense right in the middle of the point spread function, and then of course it's the dimmest at the end. There are these smaller rings around it usually, but we don't really, it doesn't matter too much. When you use a microscope with a very good objective, numerical aperture of 1.3, your point spread function is much narrower or smaller than the point spread function of an objective with a numerical aperture of 0.3. That's why the objectives is so important in the microscopes. I hope you got that from the previous presentations that Sinead and Anya presented. But yeah, the whole point is that we want this point spread function to be as small as possible. If you look at a typical image, which you've stained with many different fluorophores, this would be typically actually what it would look like. Luckily, we set the display so that we don't see these rings. We just basically see the middle, the peaks of the fluorescence. And if you put it as a function of intensity, again, you would have these peaks all around your image. A good approximation of the resolution that you can achieve is the full width here from one side to the other at the half maximum. So that maximum would be their intensity. Where that intensity is halfway, the width there, that's basically the resolution limit of your microscope or your image and your circumstances. Right, so that's just another interesting fact to remember. Basically, what we realize with a microscope is whether you can see it or not tells you whether you've got a resolution or not. Right, so the previous presentations talked a lot about different types of microscopes. We talked about wide field microscopes or epifluorescence microscopes. Many labs have epifluorescence microscopes. It's the typical ones that have fluorescence, but they're not the top of the range. There are very limited confocal microscopes in South Africa. I know of only UCT and Stellenbosch who have confocal microscopes in the Western Cape. There is also at Grahamstown and at UKZN, Wits and Pretoria, and I think there's MRC also, but there's really only one confocal microscope per institution often here in South Africa. So your wide field microscopes are the ones that you might have in your lab. The difference between them is basically that you would see much more detail with a confocal, and you can actually look. You do optical sectioning. When you do a course that we will present in a few months on confocal specifically, we can explain to you how we get to these types of images instead of those wide field microscope images. But just to give you an idea where super resolution comes in, from a wide field microscope, your point spread function includes all the out of focus light as well. A confocal microscope, it eliminates anything that is not in focus. The bottom and the top part here of the point spread function is excluded. So your confocal only gets the point spread function that is actually in focus. That's where the limit is. 500 nanometers in length and 200 nanometers in width. That's it. With super resolution microscopy, if you look at that same point spread function that's in focus, and we present it here, this comes from current protocols in toxicology, but this is a typical representation of point spread functions. A confocal might look like this, your point spread function. If you deconvolute, if you do deconvolution, you can reduce that point spread function quite significantly, but still it's not as good as it can be, as good as we would like it to be. So there are these three types of super resolution I've mentioned. The first one is called SIM, Structured Illumination Microscopy. And you can actually, today there are ways to do 3D structured illumination microscopy. So from a 2D SIM, point spread function is already reduced in the X and Y axis. And if you do 3D SIM, you also reduce the resolution in Z, in the Z plane. The second type, we don't have this STED type. You reduce the X and Y resolution significantly. It's about 30 nanometers, where this is about 200 nanometers. However, the initial STED microscopes couldn't resolve in the Z plane. Today they can. The 3D STED and 3D STED with deconvolution brings down the point spread function quite significantly to very, very small, to a very small point spread function. And then the type of that we also have, sorry, is the palm storm type of super resolution. If you use it in 2D, you again don't really resolve it in 3D. You only really bring it down in the XY plane. But with 3D palm storm, which we also have on our microscope, you can bring down this point spread function significantly again. Right, so what I will talk about, we have the, which I've put in a block here, we have SIM, and we also have the palm storm features on our microscope. And I will now discuss these two features in more depth. So, no, not yet. I will first quickly tell you what the three different types of super resolution is about. On STED microscopy, the one that we do not have in Stellenbosch, you need two types of lasers. You have an excitation laser, and that excitation laser must be the excitation laser for your fluorophore. If you are using FTSE or Alexa 488, it needs to be a 488 nanometer laser. A STED laser is another laser, and in some way, which is very physics based, they managed to get this laser into a donut shaped, as you can see here, laser. And that donut shaped laser is going over or scanning over your sample with the excitation laser. And the wavelength of this STED laser is of some, and the energy coming from that laser is exactly enough to deplete, we call it depletion, some of the wavelengths and light on the outside of the point spread function from the excitation laser. So your excitation laser, instead of having the full width here, the outer parts of the laser is depleted so that there's no fluorescence there, and the actual laser beam that reaches your sample is basically much smaller. You get the inside diameter of the donut-shaped STED laser, which then resolves quite a lot. I am not very familiar with all of this, so if you have questions, you need to send it to me and I will find out the correct and accurate answer. But we deal a lot with XR-SIM and localization microscopy. So SIM is a physics, is a type of microscopy where you can prep your sample fairly similar to what you would have done with confocal, but instead the way the microscope is set up, you acquire the images, enough images to, with algorithms and processing afterwards, you actually get the resolved image after processing. And I will explain how it works a little bit later. Localization mapping microscopy has more to do with your sample preparation. It's where you stain your samples with fluorophores and into a buffer, which allows the fluorophores to blink. We call it blinking. They switch on and off at different times. They are not all on at the same time. And because they are on at different times, we can map and localize each fluorophore based on its point spread function. I will also go into this a little bit deeper later in the talk. But basically, this is the 3 types that's commercially available. Next one. So let's talk about structured illumination. When you put a sample in, now I want you to imagine that this is a sample of muscle. It's the type of sample I know best that have striations. This is not something physical, this is your sample. If you overlay in the beam path already something that has a very predictable structure like this grid pattern or grating, if you overlay that over your sample, you see a new pattern. You can see there's stripes on this side that's quite defined, and then there's these finer patterns on this side where your sample already have finer structures. So you have new patterns. If you put a different type of grating over, the pattern changes slightly and you get a different pattern. These patterns we call Moire Fringes. If I can show you some examples, this is where, probably in a zoo somewhere, where you look through the fence, and you can see there's all these patterns on the fence. They are called Moire Fringes. Here on the right hand side is more theoretical. If you have some sample with specific features like this wobbly piece or a nice, you can see there's something going on there in the circle, and you put a very structured, predictable structure over it, you get a whole new pattern. And this pattern is much more defined or much easier to work with than the original sample with just a few wobbles. And the same with the last example. So how does it work then in SIM? What the system do, the processing part of the in SIM, is to create a Fourier transform of your image where you've taken the images with a grid pattern over. I've taken out slides, I just realized. When we image SIM images, we have a sample, and in the beam path we put a grid pattern, and we rotate that grid pattern, and we take many images, at least 5 images per rotation, and we can rotate the grid pattern 3 times or 5 times. So in the end of just one plane of imaging, we get 25 images or 15 images, and that's only of one color. So we can change the colors. Then you get, we have 4 color SIM, 405 nanometer laser, 488 nanometer laser, 561 nanometer laser, and a 614 nanometer laser. So we can do 4 color SIM. Of each of those, we can do Z stacks, and those Z stacks of each plane, each color, it can take 15 images. And then the processing works. In the processing part, a Fourier transform is created based on all those many different images. Now, a Fourier transform is something that I myself doesn't really understand very well, but there's a very nice YouTube photo that I've put this slide on here so that you can see how complicated it is for a biologist. Physics people will understand this fairly easily, but as biologists we don't really need to understand it, but it is there to, if you would like to go and view it. But basically it represents the frequencies of your light, depending on where the frequencies are, high frequency parts of your image and low frequency parts of your image, in a whole different way, where your high frequencies are represented on the outside of the Fourier transform and your low frequencies in the middle. Low frequencies are usually the ones that are not very well focused and not resolved very well. So in the processing, eventually you would leave out the middle part where the low frequencies are. Also, the grid pattern forms a very nice pattern on the Fourier transform, and based on the fact that it's a known predictable pattern, we can also exclude where they are. But what is then left is much more resolved images, than what you had before you've processed the Fourier transform. These are some of the examples we've done. This is red blood cells infected with malaria, where you have ring stages. There's a ring stage on the left-hand side. You have trophozoites, and then schizons. There's also schizons, and then right at the end, before the red blood cell would burst open, it will form merozoites, and they would then expel their little the parasites to infect new red blood cells. So this was one of the nice projects that we did SRSIM on. We've done quite a few already. I just don't have all the examples anymore or in the presentation. This is Mycobacterium tuberculosis. When you stain it with Nile Red and Auramine O, you can actually see small lipid bodies in the bacteria. If you don't have super resolution or SIM, you just see blobs of green over red, where with the super resolution now we can actually resolve small droplets and we can physically go and count how many lipid droplets does the bacterium have, which is very, very useful if you are trying to find treatments against the Mycobacterium tuberculosis. We've got a few papers out. I'm not going to go into too much detail, but then this one here was quite interesting. We can also use super resolution on polymer samples. These are little microcapsules where they form with different monomers, and the first monomers were, in the top example, were green monomers. Eventually they added non-stained monomers, and lastly they added red-stained monomers. And if you see here on that image there, there's actually a gap between the red and the green. You will not see that gap with normal confocal. It's only with super resolution that you can actually see that there was some layer in between the red and the green layer. And then they did the same thing the other way around. Red first, then green, and the overlay. Right, so this is SIM. And the next part I would like to talk about is the third type of super resolution called SIMLIM. Now we started, when we bought a microscope, we talked about Storm and Palm, but they are talking specific about their very specific sample preparation. Collectively, they are all basically a localization mapping of single molecules. How we reach that level of single molecule localization depends whether you can do photoactivated localization mapping or whether you are depending on stochastic optical reconstruction, STOR. And both of them has to do with nanolocalization of the signals from the fluorescent dyes or the proteins that you are using. This diagram from the ZOYC, I think this is the ZOYC campus, is to me one of the best ways to show what it's about. In 2D, your fluorophore would have this point spread function quite wide, as you can see, and we talked a lot about point spread function. But as we know, if this is circular, then we can determine where that fluorophore should be. It should be right in the middle of the point spread function. So instead of looking at the whole point spread function in a circle, we could say that that fluorophore is right in the middle. The problem with resolution comes in if you have thousands of molecules in your sample, your point spread functions start to overlap and you don't only have nice round circles anymore. And that's the problem with localizing it from a confocal image. So now the solution is not to have all those thousands of molecules on and fluorescing at the same time. So our chemistry partners in microscopy, they've developed ways in which they can switch between an on stage and an off stage of a fluorescent of a fluorochrome. The idea would be that you switch on only some of the fluorochromes at once, and they would each have their circular point spread function, and the microscope would record their position. Eventually they need to switch off, and another set of fluorophores need to switch on. Just quickly want to see if I have it on a slide here. Yes. You will see there, this is the blinking effect. Not all fluorophores are on at the same time. So because you are doing a recording, each little circle will be recorded and can be used to localize the fluorophore that caused that point spread function. And right at the end, if you can see here, this on the left hand side is the point spread function map On the right-hand side, taking all those point spread functions out and only localizing the fluorophores, you have a much better resolution. So the way of imaging, you would have your, let me just quickly see, you would have your sample. In some cases, you'd have a lot of fluorescence in the beginning, especially in storm, but eventually you switch all of the fluorophores off. And then sequentially, you start switching on some fluorophores, this one and that one first, to the point where they are switched off. Eventually, these two would switch on, then those two, eventually those two, and right at the end, you would have a localization point for each of the fluorophores that was on during your time, during your video. So as you can see, single molecule localization mapping of SimLim depends on a time lapse. You need to do a full time lapse of the blinking to be able to do bomb storm. So at this stage, it's something that you need to do only on samples which are really, really stable. You can do it on live samples, but the live samples, the proteins that you are interested in should really, really be non-moving at all for the duration of your time lapse. Right. Let's see what I've had lost. Okay, so this is again your point spread function of a fluorophore. You usually, because it's a nice full circle with a peak, we call this a Gaussian distribution, as you would probably know. And then you can determine right there in the middle the middle point. But as you also can see, that circle doesn't look nice in a pixelated image, so we can't. It is not completely accurate to the 0 point. We can basically determine it to either 10 nanometers or 30 nanometers accuracy, depending on how well we can represent it in the pixels. Basic principle behind the technique is that the position of a single molecule can be localized close to 1 nanometer or better if enough of the photons are collected and there are no other similarly emitting molecules closer than 200 nanometers. Now, like I said, the 1 nanometer accuracy is probably theoretical, because our pixels, we almost never image anything with a 1 nanometer. No, it's not possible to image your samples with 1 nanometer or 10 nanometer pixels. Otherwise, you would not be able to store those images. You usually have about 100 nanometer, 100 to 200 nanometer pixels, and those to resolve a 10 nanometer or a 1 nanometer fluorophore in a 100 nanometer pixel is still just a calculation. So the more photons you have, the better you can estimate where that molecule is within the pixel. Right. So in terms of the type of dyes, each dye has different properties. For example, a Sci5 photo-switchable dye has a bright stage and a dark stage. And depending on some sort of input, they can switch between these dark state and bright state. I will put it on another slide, what we do to actually instigate that. Sometimes it's a typical light. Red light or blue-green light can make them switch. And it's usually some switch in the molecule's conformation. This one here at the bottom is photoactivatable rhodamine derivatives, which can switch due to UV light. We usually use a 4 or 5 nanometer laser to cause this switch between a dark state and a bright state. Dronpa is a chromophore in fluorescence proteins, which can also be used to switch from dark to light. This is a typical principle of POM, which is a photoactivatable light or localization mapping. Here you start with dark. None of the molecules have fluorescence at all. If you use UV light or 4 or 5 nanometer light at a very, very low intensity, you can only activate some of these fluorophores. It's very important that your laser must be low. It should not be a higher, otherwise you will switch on all of the fluorophores in this area. Now, if they are switched on, they're not fluorescing yet because you're not exciting with the right laser yet, But if you at the same time then switch on the correct laser for excitation, you would make these three that was activated actually fluoresce. None of the other fluorophores will be excited because they're not in an activated state. Eventually those three will fluoresce, you record it, and they would photobleach or switch to a dark state. They would not switch on again for the duration of this experiment. But because you have your UV laser again, low intensity, some other fluorophores would switch on and you would excite them, record them, etc. Right. Then, now not all fluorophores are exactly the same. If it's just a photoactivatable dye, you would have them in an off state. They would not be fluorescent. With UV light or some other way, mostly UV light, they would go to an activated state. And then when you use your correct laser for excitation, they would fluoresce in the color that you would expect. And eventually they would bleach. You also get some you get some type of fluorophores that can switch between dark state and light state. This would be useful when you do multicolor fluorescence, because when you image one color, that UV light is actually activating every color. It doesn't specifically activate only the red dyes that you have or the green dye that you have. It will activate everything. But if you are able to switch everything that was activated by accident off again, and you reactivate, you can now you can first go for one color and then go for another color in the next round. But eventually all of these would also bleach. A very useful fluorophore would be one that can do photoconversion, usually reversible, if possible. But the principle is that you actually have a fluorescent marker. I can't remember the one name now that's very important. I can't remember, but it starts as a green fluorescent fluorophore. But then when you put UV light on, it changes to a different color. And that change is only in some of the fluorochromes at a time. So you would start off with, say, for example, a green dye, the whole image would be green. And then if you switch to the red part where you have UV activation, only some of them will change to red. And you can image the red in the palm storm feature, eventually switching all of them from green to red. And you would then have a red a super resolved image in red. Why this is useful is if you don't have any color, for example, in the first two types, it's very difficult to decide where to focus properly. You need something to actually focus the sample on. A normal bright light doesn't allow you to finely tune your focus. Where if you have a fluorescence marker that is already fluorescent, but you're not activating it at all, you can find the best focus possible and then switch to UV, switch to blinking of the red dyes or the red color, and eventually you have a palm storm image in red. But you were able to focus properly because you had fluorescence from the start. One last type is where you have a fluorescent marker, which is what they call caged. I think it's a conformational change. The UV light causes that conformational change and it switches on. And again, you can image a blinking experiment. Oh, I'm not that far away from the end. But bear with me a few minutes more, please. Right, so now if you can imagine, we are imaging these images more or less 10 minutes at a rate of about 30 nanometers, 30 milliseconds per frame. I remember, yeah, no, not 30, 13 milliseconds per frame. So your frame rate of 13 milliseconds, you don't have time to do a Z-stack because you want to get in that plane of focus. You would like to capture the next image and the next image and the next image, all in the same plane because the blinking is constantly happening. There is no time for a Z-stack. So how do you determine the three-dimensional position of that fluorophore? You are not changing focus, you're not doing a Z-stack, but in some way you would like to know the spatial, the space in 3D where that fluorophore sits. So the very smart scientists worked out a nice way to do this. Let me show you. Right, if you put into the beam path something they call a phase ramp. This is the long name for 3D POM, phase ramp imaging localization microscopy. But you put this phase ramp into the light path. It basically, I just want to see if I have that slide in. Yes, it comes from photography where you use a phase ramp where if something is out of focus, it is an it gives you a little bit of an offset, but if something becomes in focus, there's no offset anymore. So if I go back to this one, if you put this phase ramp, it's basically two pieces of glass and they are not completely 180 degrees, they're stuck together with glue and they are at one angle, there's an angle of 179 degrees between the two. And that offset of 1 degree allows for a new point spread function to form. And this point spread function is consisting of two parts and they twist a little bit around each other. So they have a very unique shape, this new type of point spread function. And if you can see in Z, they don't look the same all the way through from top to bottom. So if you would have looked at images of them cut through that Z stack, you would have seen that this shape looks different from that one, this one, this one, and this one. And what you would usually do with a 3D POM is you take your beads. We have calibration beads. They are circular or round or spheroids. If we image them, we can actually see what the shape is of the point spread function using the phase ramp in a Z stack. Okay, this is where you don't have blinking or anything. You just image your bead with a phase ramp in the light path, and you get this shaped point spread function. Then you use this point spread function shape as a standard back in your software. And when you image your sample, let me just get to the next one. It is supposed to be a video. I'm not sure if it's going to show. This is the phase ramp. It goes underneath your objective. It just inserts underneath the objective and cause that point spread function to split. Ah, there it goes. Okay. So once you put it in, each point spread function, this is two. Just quickly see if I can get it. These are two different fluorophores. When you put the phase ramp in, they split each one in two parts. They are actually at a 45-degree angle because our phase ramp is 45 degrees. And then if you take a Z stack, here on the right-hand side, black one. If you take the Z stack, they should change shape as they go through the Z stack. So you'll have far above the focal plane at 700 nanometers this type of shape, and all the way down below 700 nanometers you would have this type of shape. Eventually you can draw a standard curve of what it should look like at different levels above and below the focal plane. And then this is like the new point spread function, as I said. In your image, if you have a flat plane, you would have some of the fluorophores that have a circular, a nice 45 angle in focus shape, but other fluorophores will have a different shape depending how far above or below the focal plane that fluorophore sits. And based on that shape, we can then map and say, right, if they are far above with that type of shape relating to the 700 nanometers above focal plane, we will color them red. And if they are 700 nanometers below the focal plane, we will make it a blue color. So this is basically a heat map based on the Z position of the fluorophore. Not really the color of the fluorophore, but actually the position of the fluorophore. And this is where we get this is from our beads. We've imaged that on our microscope. So you can see at the nice 45 nanometer angle. Our 405 nanometer laser is not particularly bright because we are using it as an activation laser. That's usually why our 405 nanometer laser images don't look as good. But as you can see, our far red lasers look fairly good. And that's an imaging of the 3D resolution. I'm going to stop there when I give you the presentation, there will be extra slides with recommendations, etc. But I don't want to take this presentation too long. Right, I'm quickly going to look if there are, if I can spot. Yeah, no, I don't see specific questions. If you want to ask a question, you're welcome to unmute yourself and ask a question, and I will try and answer. No questions?
Lisa, there's a question about bleaching.
Okay, where I am trying to scroll back, I don't see it. I just see, I can see your screen and things like that.
Yeah, so the last input in the chat area was from Sahid asking, what do you imply by bleaching? Okay.
This is a general term, and it's basically, sometimes it bleaches completely. Bleaching means every fluorophore is light sensitive. Sometimes the light, whether it's UV or your laser light, and even in normal confocal imaging, you need to be careful of bleaching. You don't want to change your fluorophores to a state, a chemical conformation, or they actually get damaged by light. So they change in some sort of way where they don't fluoresce anymore. They are not capable of being excited by laser and fluorescing anymore. But in this case, bleaching would mean that they are so completely damaged that they would not fluoresce anymore. But that is slightly different from the dark state. The dark state can often be changed back into a fluorescent state as well. I hope that answers the question to some degree. Right. I also saw something here. Could you re-explain the Z point or Z plane? Right. So in terms of, I'm going to explain it in terms of the point spread function. When you have a circular or nice round, more or less round fluorophore, that round fluorophore will not look like a spheroid on a confocal microscope or any light microscope. It will look like this elongated shape. That's just how it will always work. The limit of resolution, the way it's excited, the light is, it will be elongated in the direction where the light is coming from. So when I talk about the Z plane, I'm talking about the elongated part of the point spread function, where the X and Y is the shortest plane in from left to right. I'm not sure if Ms. Raz meant the Z point in the Palm storm, but that was Z position in the point spread function means the elongated part instead of the nice smallest diameters. Z plane in Palm storm again means depth, how deep you are into the sample, or no sample, no matter how thin you section it or how thin your cell, if you have a monolayer of cells, you would always have some sort of depth, and that depth we talk about is the Z dimension of the sample. Okay, anyone else with a question? Are there opportunities for in-house on microscopy? Right. We usually do all of this training in-house, but due to the pandemic, we are not allowed to have any more than one person in the microscope room. So we are not doing physical or training in front of the microscope at the moment. As soon as we can actually allow more than one person into the lab, we will again, or not into the lab, into the microscope room, we will again start with in-house training and showing you how to use the microscope by yourself. But at this stage, it's only online training due to the funding. Anyone else? I see a hand here. I'm not sure whose hand it is.
It's my hand.
Yes.
I quickly want to know, in terms of most of the images that we've done or that I've looked at was confocal with Z stacking to see if we can find if there's any interaction between the two molecules that I was looking at. Mm-hmm. And then in the Z stack, we can actually see if they are overlapping, if they, occupy the same space.
Yeah.
In terms of the palm imaging or the spatial orientation with the 3D palm, would that also be a possibility to track the depths if we struggle with the Z-stack, to see more or less where they lie and give different colors to the different fluorophores or not?
I'm going to show you one slide quickly. This one. You would need to get your sample down to about 1.4 micrometer. The 3D palm is limited in the way that you can really only resolve it from a 1.4, 700 nanometers above and 700 nanometers below the focal plane. Your samples, I know, are unfortunately too thick. So if you can get them down to 1.4 micrometers, and why I'm saying 1.4, you don't want any other fluorochromes or molecules or anything above the focal plane or below the focal plane to distort your images. The important part is not to have a lot of noise. You've heard a lot about signal to noise ratio in the previous talks, and signal to noise ratio makes it very, very hard to get to these fantastic images. So you always have to try and get rid of noise, and in your case, the thickness of the sample above and below what you want to see would make it very, very difficult to get rid of the noise. Does that answer your question?
Yes, it does. Thanks, Lisa.
Right, cool. I saw, yeah, are we having certificates? I'm not sure. I'll have to find out from the organizer of this whole online training, because the microscopy training is not the only webinars we have. We had webinars on mass spec and electron microscopy, etc. I'm not sure if we're going to have certificates. We are actually. I've almost forgot to download the attendance register. I'll do that, and so I'll know whether you've attended or not. But I don't know whether we are going to give certificates. Are there any other questions? Don't see any hands. Yeah, so now's your chance. Otherwise, I'm going to say thank you very much for joining again. On Wednesday, I think it will be a lot again about your own images, what you can do with your own images. And if you do have some questions, please don't forget to email me the question. Give me some background of what you've imaged and what the problems are you have, or just in general what you've been thinking about, and I'll incorporate that into Wednesday's presentation. Right, thank you for joining, and may you, in terms of recording, I will also save this recording and send it with the recordings of the discussion groups on Friday. Hopefully I will work on the discussion groups of Friday first, and then I'll do this one and share it with you. Okay. Thank you very much, everyone.
Thank
Goodbye.
you.